Welcome to this video on the biochemistry of congenital adrenal hyperplasia or CAH. The most common cause of CAH is the mutations in this 21-hydroxylase gene which disrupt HPA regulation. In this video, we will explore the adrenal gland and its regions where steroid hormones are made. In addition, control of normal HPA axis and its disruption in CAH, biochemical basis of CAH, its inheritance patterns, symptoms, and current treatment approaches will also be discussed. Before we discuss how HPA axis is regulated, let us first locate the adrenal gland and understand its region where steroid hormones are made. Adrenal gland is a small triangular organ that sits on top of each kidneys. Its outer part is called as adrenal cortex, which has three layers: zona glomerulosa, which makes aldosterone; zona fasciculata, which makes cortisol; and zona reticularis, which makes androgen or sex hormones. Let us look at the regulation of HPA axis. Under normal condition, hypothalamus releases CRH, which stimulates anterior pituitary gland to secrete ACTH. ACTH acts on the adrenal gland, increasing the production of cortisol. Cortisol in turn provides a negative feedback regulation of the HPA axis to keep the system balanced. Thus, cortisol synthesis is regulated in the zona fasciculata of adrenal cortex by this axis. Let us discuss the HPA axis regulation in CAH. In CAH, cortisol concentration is decreased significantly, which disrupt the feedback regulation of HPA axis, leading to a consistent increase in ACTH, which causes adrenal hyperplasia and excess production of androgen by the adrenal cortex. This causes abnormal development of external genitalia, including clitoral hypertrophy, resembling the penis but ambiguous. However, the development of internal reproductive organs such as ovaries, fallopian tubes, uterus, and vagina is normal. So let us discuss the biochemistry of CAH. In this section, I will focus on three aspects. First, why cortisol is low. Second, biochemical basis of the disease. Third, what is an overall derangement in the steroidogenic pathway leading to the symptoms. There are three regions which are shown here: zona glomerulosa, zona fasciculata, and zona reticularis, which are responsible for the synthesis of the steroid hormone. So let us start with zona glomerulosa. Cholesterol is brought into the mitochondria by star protein, where it is converted into pregnenolone by the action of SCC or side chain cleavage enzyme. Pregnenolone is converted into progesterone by the action of 3-beta HSD. Progesterone is subsequently converted into deoxycorticosterone by 21-hydroxylase enzyme, and deoxycorticosterone is converted into corticosterone by 11-beta-hydroxylase enzyme. Corticosterone is finally converted into aldosterone by the action of aldosterone synthase. So this is to fix our electrolyte balance, and this is produced in the mitochondria. Now coming to zona fasciculata, where cortisol is synthesized, the pregnenolone is acted upon by 17-hydroxylase enzyme, producing 17-hydroxypregnenolone, which is converted into 17-hydroxyprogesterone by the action of 3-beta-HSD. 17-hydroxyprogesterone is then converted into 11-deoxycortisol by the action of 21-hydroxylase enzyme. 11-deoxycortisol is converted into cortisol by the action of 11-beta-hydroxylase enzyme, and this is for the regulation of metabolism and produced in the mitochondria, also in the zona fasciculata of adrenal cortex. Now CYP17 also has second activity, which cleaves the side chain, and as a result it produces the dehydroepiandrosterone, which is called also known as DHEA. This is an androgen, This is converted into endostenedione by 3-beta HSD. So these are endogens. Endostenedione is then converted into testosterone by 17-beta HSD. Testosterone is subsequently converted to DHT or dihydrotestosterone by the action of 5-alpha reductase. So what is the biochemical basis? Biochemical basis is the mutation or deficiency of 21 hydroxylase enzyme. As a result of that, the concentration or the aldosterone and cortisol are depleted, whereas there is corresponding increase in the upstream substrate, which are funneled into the zona reticularis, where there is synthesis of the sex hormone takes place. There is a high level, there is an excess of androgen production, which leads to the manifestation of symptoms. Inheritance of CAH. The gene CYP21A2 encoding 21 hydroxylase enzyme is present on the short arm of chromosome 6. This is an autosomal recessive condition which affect both male and female children. However, the symptoms are more severe and devastating in female child, leading to mutilation of the appearance of external genitalia. These patients are associated with some symptoms of salt wasting such as hyponatremia, hyperkalemia, hypotension, hypovolemia, which are due to low level of aldosterone in these patients. Symptoms such as hypoglycemia, vomiting, and fatigue are associated with the decrease in cortisol level, and the symptoms due to abnormal appearance of external genitalia are due to increased or excess of androgens in these patients. These patients are managed by aldosterone and cortisol replacement therapy, and external genitalia are corrected by surgical operation. Congenital adrenal hyperplasia is more common in Eastern Mediterranean and Southeast Asia than in Western countries, with a global incidence of approximately 1 in 9,000 live births. Thank you very much for watching this video. Please like it, share, and subscribe it. And until next time, take care. Bye bye.